Well, hey AP, hope you're doing well when you get this watch this video. I'm sorry if you've been sick. Being sick is so hard. And Jeremiah 17:14 says, "Heal me and I will be healed, save me and I will be saved, for you are my strength." Of course, he's talking to God there. So healing comes from God. Believe in that, praying that for you. Okay, so just want to put a face with a PowerPoint presentation just to kind of show you that you are not forgotten and that you are around. This is also to help those who were in class and maybe were distracted or maybe came to class that was really who were struggling with sickness and feeling poorly. I'm going to go over chapter 5 for you, and so let's go ahead and jump into it, okay? So chapter 5 deals with cellular membranes, okay? And let's move this over some. And so cellular membranes, we talked about caffeine and how does caffeine affect the body. And so we talked about how it really mirrors adenine right there, and so it acts as a blocking mechanism. Okay, so we'll skip all this because we talked about all that. Remember, marijuana debate's coming soon, so be ready for that. And so I'm just going to really go over the top kind of concepts in this video for time, of course. But you can see right here we talked a lot about the synapse and the unlucky Chucky worksheet. And again, cell membranes, your body communicates, and it communicates through cell membranes. So you can see all these neurotransmitters here. And based upon what this neurotransmitter is and being able to have it bind to a receptor protein acting as a ligand, it really changes how we feel, changes how we think. Some neurotransmitters are responsible for letting our brain and muscles communicate, therefore letting us live, of course, and move and breathe and stuff like that. So before beginning this chapter, as I said a while ago, I would pause here and make sure you can do all these 5 bullet points. And so the basic model in science for the plasma or cell membrane is the fluid mosaic model. And so these phospholipids are like a lake, and the proteins are floating in it. Okay? And so right here you can see that this is extremely important. So here's the lake, right? Remember, this is charged right here. So you have your polar head that's charged. So that's hydrophilic. And then you have an entire phospholipid right there. And this is the hydrophobic section, right? And then you have cholesterol, which affects the integrity of a membrane. You have proteins. Sometimes they're peripheral proteins, which means on one side or the other. Sometimes they are proteins that have a carbohydrate on the outside. So that's called a glycoprotein. And glycolipid would be right here, okay? Those are really kind of signaling or cell recognition type situations. Extracellular matrix, what I'll talk about at the end of this video. Some membranes go across it completely, integral membranes, transmembrane proteins. You can have the cytoskeleton right here. Sorry for the circling there. And so I would know, let's do a different color to make emphasis, but I would know this slide, okay? I would know it very, very well. I would know this picture very well and understand the functions of all these different components. Okay, that would be extremely important. And so the plasma membrane is common to all cells, and so it separates the internal from the external, and it does this by having that phospholipid bilayer where the tails are this nonpolar hydrophobic part and the heads are the hydrophilic part. Okay, so hydrophilic regions are charged, the heads, hydrophobic regions are the tails. That's a picture of a membrane. And so you can see right here, I need to adjust this again, sorry. Oh, no, let's just go a little smaller. Pause this. Sorry, I had to adjust the screen here a little bit. But you can see right here, so bilayer is formed, of course, bi meaning two and stuff. So here you can see the two phospholipids, the tails joined together, the hydrophilic heads point towards water on the outside and the inside. And so there's different components. The lipid component, of course, is a phospholipid bilayer. Protein components, they float around like icebergs or on a sea. And so there's membrane proteins, peripheral, of course, on one side, integral or transmembrane on the other. And then cholesterol affects the fluidity of the membrane. So they shift about. This shifting can be very rapid. So I talked about in class this experiment where they radioactively labeled mouse proteins and human protein, then fused the cells together, probably using electrical shock. And after about an hour, you can see all of them have been mixed up, they've moved around in the floating sea of lipids. And so as temperature is cool, here's the thing. Membranes switch from a fluid state to a solid state. Of course, losing energy. The temperature at which a membrane solidifies depends upon the types of lipids it includes. So here's the thing. A membrane remains fluid to a lower temperature if it is rich in unsaturated, which means there's, saturated is the straight lines, and then unsaturated is the kink, right? So there's space there. And so being too tightly packed together will cause it to freeze more. And so you want the space when cold temperatures. Cholesterol has a different play. Cholesterol restrains movement, so it keeps it together. And at cool temperatures, it maintains fluidity by preventing tight packing. So membranes must be fluid to work properly, and they're usually as fluid as olive oil. Okay, so here's just an example of that. So in a very warm environment, you would want less unsaturated, more saturated. And so there's cholesterol. So two important factors: lipid composition, which is unsaturated or saturated, and temperature determine membrane fluidity. Okay, we've already talked about peripheral and integral membranes. Here's just another picture. You see right here the integral membrane, cytoskeleton, peripheral membranes, hydrophilic regions, this is a phospholipid, such so on and so forth. And so a transmembrane protein extends through the bilayer on both sides. It could possibly have different functions in its external versus the transmembrane domains. So you can see right here, and then think about charges, right? I would encourage you, okay, these amino acids probably have to have a certain charge. These amino acids have to probably have a certain property. These amino acids probably have to have a different property. And so the next region that we talked about that I want to talk to you about is membrane proteins. Membrane proteins do a lot of different things. Okay, so channel proteins allow passage. Carrier proteins literally carry a membrane across. That's kind of important with the glucose, so it assists passage. Cell recognition, yeah, your body knows you from not you. Crazy important. Moving on, you have your receptors, which allow a cell to respond to signals, of course, because they receive a signal, a ligand. Enzymatic proteins, of course, carry out metabolism, anabolic or catabolic reactions, building or breaking. And then junction proteins attach adjacent cells. Okay? And so here's the 6 major functions. Again, knowing all this stuff about proteins is crazy important. I would start that and then figuring that out, okay? Let's go back here, sorry. And then this is just a nice picture in the notes as far as kind of all those functions go, okay? Cells recognize each other by binding to surface molecules, those glycolipids or glycoproteins. And so membranes have distinct inside and outside faces. And then how does this happen? Well, it happens in the endomembrane system. So you're packaging proteins here, attached carbohydrates, transmembrane glycoprotein, you can see right here. And if it's on the inside of the vesicle, it fuses, and therefore now it's on the outside of the cell. If it's on the outside of the vesicle, it fuses, and there it is on the inside of the cell. So again, understanding how this works would be important. And let's go on to part 2. So 5.2 is really all about passive transport, okay, and hypertonic and hypertonic. So passive transport requires no energy, no ATP. Active transport requires ATP. Thinking about downhill versus uphill, right? If you're riding a bike, it's going to require so much ATP because you're going from low to high elevation. You just coast, have a good time. It's beautiful scenery there. And so you can see that this is simple diffusion, facilitated diffusion, or active transport. So this would be diffusion, high to low. This is active transport, low to high. Okay? And so simple is just straight through the bilayer. Facilitated needs help. Okay, so again, knowing what molecules readily cross the membrane would be important. So polar molecules, charged molecules, they do not cross the membrane easily. Even really water needs help getting across the membrane. And so transport proteins allow these hydrophilic substances to cross. The channels, they have specific, designs to allow molecules to cross. Aquaporins, of course, is water channels to allow water to cross. Carrier proteins bind and change shape, but yet still no energy is being used because we're going with the concentration gradient. Okay, so diffusion is going from high to low. Okay, greater to lesser. So here's a picture here. So you can see high to low, straight through the lipid bilayer. So these are like small gases, nonpolar molecules. Facilitated diffusion needs some help. These are like polar molecules. Again, charge is more important than size, right? Charge is more important than size. Ions would need help across. But you can see this is low to high. So we're going uphill there. We're going against this concentration gradient. So we need help. Okay? And so speed of diffusion depends upon the diameter. Of course, small molecules go faster. Temperature, more energy would be faster diffusion. Concentration gradient, if the hill is steeper, if we're going from like a ton on one side of the membrane to not that much on the other side, that would also increase it. Okay? So O2, CO2, small nonpolar lipid-soluble molecules, they readily cross. Okay, amino acids, sugars, ions, even water, they're hydrophilic. They need some help. They need help getting across. Okay. Again, no energy is expended. Osmosis is the diffusion of free water. So water goes from high freedom to low freedom, or it goes from low solute concentration to high solute concentration. This is going to be super important when it comes to our lab, which I know some of you missed, some of you did, but that's going to be super important when it comes to that and trying to understand and order those sucrose solutions in order of molarity. And so that will be important when it comes to the lab. So you can see right here, here's going from low salts to high salts, and water would go across this membrane. The salts cannot cross it until it reaches an equilibrium of equal movement. And this leads us to some vocabulary tonicity. And so you can have isotonic, which is the same. So water equally goes in and out equally. There is no like stoppage of water movement. It's just no net movement, which means no side gains anything. Hypertonic solution, the cell loses water because water leaves the low salts in the cell and goes to the higher salt concentration outside the cell. And it's the opposite for hypotonic. The cell would gain water or possibly even burst due to gaining so much water. And that's, again, how some people have died drinking too much water. Their brain tissue swelled. And it's really tragic, to think about that. And so here's just a picture kind of illustrating those points if you want to pause and kind of think this through. Plant cells are turgid and generally healthiest in a hypotonic environment. So we have a lot of water just rained. That central vacuole fills with water and pushes on the cell wall. Flaccid, equally, but not the best. And then plants are dying if they're in a hypertonic solution. Like if seawater, salt water is leaching into soil, and if the plants don't have the adaptations necessary to deal with that, struggling to survive. Okay, and this relates to osmoregulation. The paramecium, right here, it lives in a hypotonic environment, so water's always coming into the cell, so it can't survive like that, but it has a contractile vacuole where it expels water from it. And then this is just another picture and an explanation of plants gaining or losing water. Okay, this can lead us to turgor pressure. A term definition where it means the internal pressure against the cell wall as it builds up, it prevents more water from entering. And so that also leads to osmotic pressure, which is an equation. I'm not going to dive into this right now, just for sake of time. We'll practice a worksheet in class. Okay, before I talked about diffusion through different protein channels, both of these are carriers and channels. They're both facilitated diffusion. Ion channels, most are gated, which means they open or close based upon a certain ligand being around. So that is important to understand. Like sometimes channels aren't open. They're not always open. Sometimes they're gated, which means they need to have this ligand around to actually open or close. Okay, so there's just a picture there of understanding that. Aquaporins, of course, water movement. Here's a carrier protein for glucose. Again, diabetics have an issue with that. My dad, of course, being a diabetic, issues with this process right here for a lot of different reasons. A lot of different reasons, type 2 reasons that this process isn't working well, but it comes down to a carrier protein. And so we practice that scientific skills activity. And so moving on to active transport. Active transport, of course, is uphill, so we have to have ATP energy to do that. We have to have the energy there necessary, okay? And so examples, movement of glucose out of the small intestine, movement of sodium out of urine and back into the kidney. But probably the most important example, that's really a staple example in the realm of biology and anatomy, is the sodium-potassium pump, okay? So sodium-potassium pump literally sets up your nervous system and many parts of your body. And so I want to go to this picture. I know this picture is in your notes, but I'm going to go to this picture right here. Make sure, by the way, this is labeled incorrectly. It doesn't go from high to high, sodium to sodium. It goes from low sodium to high sodium. So everything is active transport here, okay? So let me go through this picture with you. We're going from low, again, notice the concentration descriptions, right? We're going from low to high. So we're pumping sodium using ATP from low to high. This is sodium-potassium pump, or ATP ACE. And so then loses its affinity for sodium, pumps it out, gains an affinity, notice the shape change, gains an affinity for potassium, binds, goes through, loses its affinity, and resets, okay? Releases both. All right, so what we're doing is we're pumping +1 charge across thousands of times. So we're literally setting up an electrochemical gradient. We're setting up a charge. This membrane is positive, this part's positive, this part's less positive. And so there's a charge, there's a little voltage there. You actually have a little voltage in your body that's running with this stuff. And so it's pretty impressive. Poisons completely destroy this pump, like this poison right here, Uabane, I think that's how you say it. And it's just interesting, and this has been used, in the Lucky Tucky pack you heard about Harari, and how this stuff has been really kind of used in medicine, even in research and stuff like that. So there's two types of active transport, primary and secondary. Primary directly uses ATP. Secondary uses like what ATP has done, okay? Why is this important? Why is membrane potential important? Why is this voltage setting up important? Because literally your heart runs on this. This is your heartbeat. This is an EKG. PQRST. Cardiologists deal with this stuff every single day. If there's a lengthen here, if there's a long ST segment, some medications can actually cause a long segment there. And this is depolarization of the atria, ventricles, and then repolarization. So all this is charged, right? Because you have a membrane potential and it flips. Why does it flip? Because channels open and lets all that sodium rush in. Lub-dub, right? Heartbeat. And so sodium rushes in, it depolarizes, and then it resets it up. Sodium, potassium pump, and various other pumps. And so it's pretty impressive that literally you're running on this little tiny voltage based upon ion differences, and that's called an electrochemical gradient, okay, which is right here. Chemical, of course, the ion's concentration. Electrical is the effect of the membrane potential, okay. Plants, fungi, and bacteria set up potentials like this, but they don't set it up like us because, of course, they're in different domains of life. And so they actually have proton pumps. So you can see right here, we're still going from low to high. Protons are, of course, hydrogen ions, and so using ATP to do that is important. Co-transport is we can couple. The key word here is couple. Let me see if I can highlight couple. No, that's weird. Okay, but couple a downhill diffusion with an uphill transport of something else, okay? And so let's show you what's going on here. So here's a coupling. So this right here is primary, right? Because ATP is being used. And then we're going from low to high. And then we use this high concentration to couple the, oh, no, not a circle. My bad. Erase that. To couple, oh man, it all was destroyed. But to couple the low to high movement of sucrose, okay? And so that's how secondary transport works. And so this is just what I talked about in class, but maybe if you didn't hear it, read this. It's impressive that from this knowledge we've saved millions of lives that are possibly threatened due to chronic diseases that cause diarrhea. And so there's other ways of active transport called bulk transport, endocytosis and exocytosis. So endocytosis brings stuff into the cell. Exo releases it. Lots of different products are brought in and released via this way. The toughest one to kind of remember is receptor mediated, which means you have to have the right receptor to bring it in. Okay, and I'll show you a picture here in a second. So phagocytosis literally means cellular eating. This is endocytosis. Just another. Pinocytosis is cellular drinking, which is endocytosis again, just vesicles are forming, so they're small and they bring in fluids and dissolve substances. Receptor mediated have to have receptors, okay? And so we have to have receptors Yeah, that's better. We have to have receptors in order to work, okay? So let me show you what that looks like. You can see right here you have your receptors. The receptors aren't around. We're not bringing this stuff in, but yet they are around, and then they start folding, and the membrane starts folding and kind of pinching off and bringing it into the cell, okay? And that's just pictures of that. Exocytosis removes stuff. So we're talking about like digestive enzymes possibly, growth hormones, any sort of hormone possibly. Why is this important? Because we use receptor-mediated endocytosis taking cholesterol for membrane synthesis. But some people have a disease that they cannot do this called hyper, which means too much cholesterolemia. LDL receptors are defective or missing, which means these people are more likely to have hardening of the arteries or atherosclerosis because they don't bring in the cholesterol as much. And you can be genetically tested for this these days. Okay, so that's pretty impressive there. And then finally, our last section, 5.4, modifications of cell surfaces. And this one's the simplest, most least amount of material, I should say. And so what you have is, you have a couple of things. First off, extracellular matrix. It's a meshwork of proteins and polysaccharides. Okay? So we have different proteins like collagen, resist stretching in your skin course. Elastin provides resilience. Integrin, role in cell-to-cell signaling. We will talk about signaling eventually. Proteoglycans, which regulate passage through the ECM to the plasma membrane. So all these have a role, all these have a function. You can see the picture here. The ECM varies a lot in different parts of the body. So the bone, of course, is hard, made up most of calcium. Cartilage is flexible. The ECM is flexible. Epithelial tissue, mostly cells and very little ECM. You can see right here, there's junctions between cells. So these are proteins, possibly, most of the time involving a protein. So you have adhesion junctions of desmosomes. As I said in class, desmosomes is involved with a heart disease, them not being formed correctly due to genetics. Tight junctions, of course, impermeable, this is found in your epithelial tissue coverings. And then gap junctions allow communication via ions or signals or various other things that could also be communication, okay? And so plants, again, we often think of plants not communicating, but that's not true. Plants communicate. They have to be able to maintain homeostasis to respond to the world around. Plant cells communicate. And how they do that? They do that through plasmodesmata, okay? And so it allows passage of material between cells. It could be for communication purposes, could be for resource sharing or waste, any sort of different things. But they have a permeable cell wall because if they didn't, they would not be able to maintain homeostasis. All right. I hope this was helpful to you. Please reach out if you have any emails whatsoever. Know that you are cared for and loved by God and by your family and by us here. Yeah, let me know if I can help. God bless, guys.